using the small suction pipette on the left, and then a small needle can be inserted into a cell, and the nucleus can be removed using suction. Uh, and all sorts of fun things can happen at that point once you can get in there. So let's talk about a little bit about DNA structures. DNA, or deoxyribonucleic acid, is the genetic material that is replicated and passed from generation to generation. We know that DNA's structure is important to uh, to be able to be copied. Now, it can be copied to make a new cell, or it can be copied in the form of RNA. Its structure is made up of nucleotides, and these nucleotides are small repeating units. So in the picture on the right, we see a nucleotide highlighted here. It's made up of three main parts: a phosphate group, a sugar, and a nitrogenous base that can vary between A, T, G, or C. All right, so that A, T, G, or C and the double-stranded design was investigated by a lot of different scientists, but those that are credited with its discovery include Watson and Crick in 1953. And Watson and Crick were actually a little stumped uh, in the actual structure of the DNA molecule itself. At the time, people were not sure if DNA was a single strand, a double strand, or even a triple strand. And it was the scientific work of Rosalind Franklin, who used X-ray crystallography to actually take a picture of that DNA strand, and her image highlighted the twisted ladder that we recognize as the double helix. And Watson and Crick were able to put together the structure of that DNA molecule using her image as a guide. So there are only two ways that DNA pairs can join together in the middle. You're going to have groupings of A bonded to a T or a G bonded to a C. And there is a very weak hydrogen bond, almost like a magnetic connection, that can be separated and reformed at any time with the help of some enzymes. So the A always bonds to a T and the G always bonds to a C. Now, it is important to identify the reality that there can be mistakes in this. And this next slide shows us this. As I click through here, you'll see that adenine and guanine on the top here are both double ringed bases. Now, A will normally bind to T, and thymine and cytosine, you'll notice, are single ring bases. So in order to make it across the ladder, the step of the ladder, which is are the bases, you're going to have an A bonded to a T. That's going to give you a total of 3 rings across. G will bind to C, which can also give you 3 rings across. Now, it's not far-fetched to recognize it and have the question, well, can A bind to C, or can guanine bind to thymine? And the answer to that is yes. Errors do occur, and those types of errors are most frequent. But there is a series of enzymes and proteins that spell check the DNA to make sure that A binds with T and G binds with C, to try and prevent those errors from being passed on. So you will always have a purine bound to a pyrimidine. All right. A will bind to T and G will bind to C, and those are shown there for you. All right, so now let's talk about the actual chromosome itself. During cell division, which we learned about in mitosis, during interphase, all the chromatin, which is the loosely coiled DNA, that DNA gets separated at those A's, T's, G's, and C's right in between. They get pulled apart. And in order to copy it, there's an enzyme that fills in. So this chromosome that you see forming during interphase, or excuse me, during prophase, has already been copied. So it was pulled apart and new strands were filled in. And we'll talk about the that process in a little bit. All right. But chromosomes are able to coil up and wrap themselves around the DNA wraps itself around the proteins known as histones. Now, a grouping of 8 histones is called a nucleosome. Nucleosomes allow for enormous amounts of folding and coiling to pack that DNA in as tight as you possibly can. And right at the end of every chromosome is a region known as a telomere. A telomere is it's almost like an extra end or a cap on the end of the important genetic material, because every time that genetic material gets duplicated, a tiny little piece of that telomere gets shaved off or lost. And the telomeres can act like a genetic clock in order to regulate how long that cell can survive and have all its genetic material intact. Because every time the cell divides, the telomeres shorten just a little bit. And once those telomeres are gone, the cell will typically go into apoptosis, which is a programmed cell death. And there's some really interesting research on aging that is being done on how to prevent telomeres from breaking down and shortening. So here's a picture that you've seen before on our previous note sheet, is the coiling of DNA. And this shows the histone proteins and how they wrap around, forming the nucleosomes. Nucleosomes are groupings of 8. All right, you see the 8 histones that form this sphere here. And those nucleosomes will get coiled up, and those coils themselves will also be coiled, and those form supercoils. Now, the supercoils are folded up and packed together to make those chromosomes. The very ends of these chromosomes would be where the telomeres are. All right, so let's talk about that DNA replication process. It occurs before a cell can divide. So during replication, the two strands of DNA, which the original strands in our diagram here are in blue. So the two blue original strands are pulled apart from each other. And if you look closely, you'll see there's an A and a T and a G and a C and a C and a G. And those are all the originals. And you're going to make a complementary strand by matching up A's and T's, G's and C's, and it'll give you two identical strands. And it's done using a couple of different important enzymes. All right. Number 2 in here is identifying helicase. Anything that ends in ace is an enzyme. And the enzyme helicase is responsible for unraveling or separating that double-stranded DNA. Once those strands are separated, the weak bonds, those are called hydrogen bonds in between the A's, T's, G's, and C's. They're pulled apart from each other. There's a specialized enzyme called DNA polymerase. This is the main enzyme responsible for fitting and matching up the A's, T's, G's, and C's with its matching base pair. So that enzyme, DNA polymerase, fills in the missing material. Once those uh, bases are put back in, the two complementary strands are formed, and DNA polymerase is also going to be proofreading to make sure there's no mistakes. The DNA will coil back up, and the uh, finished copies will be released. Now, we have a link down here to take you to a video that'll show that in a little more detail. And please make sure that you take the time to watch those linked videos because they do a great job with the animation that we just don't have the ability to show. That brings us to RNA structure. RNA, or ribonucleic acid, is really only different in a couple ways from DNA. The sugar molecule between the two molecules is different. DNA is deoxyribose, and RNA is ribose. And the differences between those two sugars is actually pretty minuscule. It actually is only a difference of the location of a hydrogen and an oxygen. Number two on our list of differences, RNA never has T or thymine. Instead, there will be a U substituted in anytime a T is supposed to be there. Now, U or uracil is just a different version of a nitrogenous base, and it is found in any strand that is RNA. If you see a strand that has thymine in it, it's going to be a DNA strand. If you see a U, then you know it's RNA. That is a very important memory clue. RNA is also typically single stranded, but there are so many different types of RNA that we're going to introduce some of them. But generally, RNA can be single stranded, but it does come in some double stranded and interesting shaped molecules. RNA nucleotides are made out of the same types of materials. You've got the ribose molecule, a phosphate, and the base. But again, A, instead of binding to T, A is going to bind to U. And then G and C behave the same way. All right, so I mentioned there are three main types of RNA. All right, but there are many other types. The first three types you need to know about are responsible for helping to make proteins. The first is messenger RNA or mRNA. It carries a copy of the instructions from the DNA on how to make a protein. If you think of the DNA code as a blueprint, blueprints are always carefully protected, they're valuable, and for DNA, it's got to stay in the nucleus. But your cells can make a temporary disposable copy in the form of messenger RNA. The messenger RNA is a copy of the instructions that will be carried to the ribosome to be able to make a protein. The copy is called transcription. Now, the word transcription means to rewrite or transcribe. Transcription is rewriting the genetic code from DNA code into RNA code. It's still genetics, but you're just rewriting it kind of like as I mentioned, a disposable copy. That messenger RNA is carried to the ribosome, and there the ribosome or rRNA will bind to number two here, the transfer RNA or tRNA. tRNA will carry the particular amino acid right over to the ribosome to match up with the messenger RNA to create a growing strand of amino acids. This process of bringing amino acids to the ribosome and creating a protein strand or amino acid strand is called translation. Now, there's another memory clue here behind the meaning of translation. To translate means to change languages. I can ask you to translate an English passage into Spanish or French, or whatever language you take. That's translating it. Well, translation in genetics refers to the process of taking a genetic code in the form of DNA and RNA and converting it into a protein code, which is the amino acid sequence. So you're taking a genetic sequence and you're converting it or translating it into an amino acid sequence. DNA codes for the order and number of amino acids in a sequence to make up any protein in your body. Some of the proteins in your body are gigantic. Some of them are as small as 50 to 100 amino acids, but many are very, very large. So in order to make a long protein, you've got to have a long genetic code. The codon is a grouping of three nucleotides in a sequence to specify a single amino acid. And this phenomenon is really important to understand. So really focus right now. If you have an original strand of DNA that's underlined here in the form of these bases, AGC, GTG, CCA. This is one section of DNA. You're going to make a copy of that in the form of messenger RNA. That would be transcription. That transcribed messenger RNA code would be the base pairs that go with it. But remember, you have to apply the rules for messenger RNA or RNA production when you're making the code. A would bind to T, but you're not allowed to have T's in RNA, so you have to substitute U or uracil. So this code is going to be converted. A binds to U, G binds to C, C binds to G, G binds to C, T to A. You can always write A's in RNA, but you're never allowed to write a T. And so this last A would normally bind to a T, but you have to substitute U. This is your messenger RNA code. The messenger RNA strand leaves the nucleus, goes to the ribosome, and at the ribosome, it will match up with a piece of tRNA or pieces of tRNA. tRNA has what is known as an anticodon. It is the match to your messenger RNA, but it's done in groupings of three. So U would normally bind to A, so your tRNA code has to have the A. G binds to C, C binds to G, and so on and so forth, all the way down the line. Each one of those RNA anticodons is specific for a particular amino acid. So AGC codes for serine, GUG codes for histidine amino acid, and CCA codes for the glycine amino acid. And you may be wondering, why the heck would we need to know that, or how could we expect to know that? Well, we have a secret decoder that we're going to share with you. And that secret decoder is located on the next page. But before I get there, I just want to point out some of the other types of RNA, and this is where we really get into some of the fancy and pretty advanced technologies that we have in genetics today. So this is way beyond what's known as the central dogma. And the central dogma in biology, which you are expected to know this, I'm going to take a second and say it, but it'll come up again. DNA is used to make RNA, and RNA is used to make a protein. It goes in that sequence: DNA to RNA, RNA to proteins. But these are some of the crazy types of RNA that exist out there. The first three are the ones we've talked about. Now, rRNA, you'll see there's a word here called catalytic. All right. A catalyst is a material that helps a reaction happen. So our RNA doesn't actually carry information, but it behaves as a catalyst to help translation and transcription. Okay. tRNA is on there, transfer RNA. That's for making proteins. Uh, snRNA, or small nuclear, and I'm also going to talk about some single-stranded RNAs that are, and MI, or microRNAs, that are really important for controlling what genes are active and what genes aren't active. The RNA molecule is copied in the DNA. Here's inside of this purple nucleus, you've got a single strand of RNA produced here. This would be labeled messenger RNA. This would be, oh, my fault, apologies. Uh, this would be the RNA polymerase protein that is responsible for making the RNA. Here's your DNA strand, and that messenger RNA strand is going to leave the nucleus. It's going to travel out into the cytoplasm and move to the ribosome. That's where translation can occur. Parts of the new RNA molecule are going to be edited out. Just so you know, this process is not a finished process. There's actually some editing of the RNA where you have pieces of the RNA removed before it leaves the nucleus. Messenger RNA attaches to the ribosome. A start codon always has to come first, and that is going to code for methionine, or MET. Again, there's a decoder that I'm going to share with you in a little bit that's going to tell us exactly what each codon codes for. tRNA brings the anticodon to the messenger RNA at the ribosome, and the amino acids, or AAs, are going to be connected using peptide bonds. tRNA repeats until it just continues to transfer the amino acids until the final codon, which shows up, is a stop codon. This grouping of three bases will tell the ribosome the protein is done, snip it off, and release it. And that protein is actually going to have a pretty complicated structure. There are 4 levels of structure within a protein. You have its primary structure, which is the order of the amino acids. And that amino acid order can bend and curve and twist. We know that, or name that, secondary protein structure. This is small waves or folds in the chain. And then we form tertiary protein structures. As it folds over and bends and curves, it's going to form a three-dimensional shape. So a wave or a curve isn't necessarily three-dimensional shape. We're looking at the large scale molecule when we get to tertiary protein structure. And lastly, we have quaternary, which is the 4th level of protein structure. And it turns out that not all proteins are finished all by themselves. Some proteins consist of more than one amino acid chain interacting together. And a great example of quaternary protein structure uh, exists when you start talking about the roles of vitamins in our bodies. Many vitamins are coenzymes or cofactors for different reactions to happen. So they react or bind with other protein parts to create like a Voltron of a protein. You get this, two smaller proteins that fuse together to form one functional protein. Here's a diagram that shows this process in action. mRNA strand of AUG it moves through this blue and yellow ribosome, and through this, each one of the tRNA molecules will bind. You'll notice that tRNA has a really unique structure. Look at that shape. It actually looks like a plug. And if you think of it like a plug, it's designed to carry this red dot or this blue dot. These are examples of amino acids. They get carried over here and it plugs in and it connects the amino acid within the ribosome to the growing strand. All right. So the messenger RNA gets transcribed, it comes out to the ribosome, the transfer RNA carries the amino acids over, and the longer or the lengthening chain of amino acids is produced. This is the long chain of amino acids forming, and we would get to the last codon in here, and it will code for a stop codon. It's going to tell us that, hey, this protein is done, clip it off and release it. And I'd like to make sure that I point out this blue tag here. All right. When you're looking at your note sheet, click on this video and it'll show you protein production in action. Here's your secret decoder. Oops. All right. So this diagram is read or interpreted from the center out. There's a grouping of three bases, right, in every codon. So if I have a code that is ACU, I know that it codes for the amino acid threonine. If I have the codon GCC, I know that the code or the amino acid that will code for is alanine. So the first base in the sequence, the second base in the sequence is that middle yellow color, and the third base in the sequence is found in that narrow dark yellow band there. And the amino acid is identified on the outside. You'll notice that there are stop codons in here. And I mentioned earlier that every protein starts with a start codon, and that's always going to be AUG. Now, there can be, as I mentioned, mutations. They can be chromosomal mutations, or they can be in the actual code itself. Right? These are examples of changes that can occur to an entire chromosome. Like, for instance, in this case, the gene B is deleted. And remember, if you're deleting an entire gene, you're creating a lot of potential problems. All right. You can have duplication of a gene. In this case, they duplicate B. Now, if this B gene happens to be the gene for the production of growth hormone, then that growth hormone will be over secreted in this individual. They're going to have two sections that are going to be producing growth hormone. So this individual who has this duplication may end up being significantly taller than someone else. This is one of the ways that we recognize giantism occurring. There can be inversions. Inversions are when you take the code, which goes A, B, C, D, E, F. If it folds over on itself, they can cross over, and this is not going to be a positive experience for this individual because they're putting the genes in the wrong locations. Now, this isn't nearly as bad as translocations. All right. Translocations in this case are when those genes cross over on a different chromosome. If they cross over on an alternate chromosome, it's like moving. If you think of inversions and translocations like a book that you're looking for in the library, right? An inversion is like moving the book to a region where it doesn't belong. So if I'm looking in science fiction for a Harry Potter book, uh, and someone moved the Harry Potter book to the nonfiction, all right, and put it in a reference area, I'm not going to know to look there. And your body behaves the same way. If you move a gene from one area on a chromosome to a different area on a chromosome, it can sometimes be lost and your body won't activate that gene. Translocations take that a step farther, going with that same metaphor. If I were to take the copy of Harry Potter's book from the Central Square Library and move it to the Brewerton Library, uh, you would definitely not know to look for it there. All right. And the cell doesn't know that either. If you change the location of that gene, it becomes lost, and those genes are often not activated, especially in specific cells where certain genes are turned on and others are turned off. Here's some examples of specific coding mutations that were outlined at the very beginning of the last slide. Right here is an example of substitution. When you substitute one gene, or excuse me, one base in a sequence, just changing one amino acid will have the effect of changing one of the amino acids in the protein. So you'll see the code changed, and that made the amino acid change. This can change the folding pattern of a protein, preventing it from folding the way it's supposed to fold, and then it can deactivate that entire protein. So, one simple mutation, changing the bend or fold in the protein because you have a different amino acid there, can make the amino acid or the protein non functional. Here is another example, but in this case, this is an insertion. Now, insertions and deletions are particularly dangerous because they change the reading pattern in the DNA sequence. All right, so we call this frame shift mutations. When you insert an extra an extra base, you push everything out of alignment. So the code gets shifted because it's still going to be read in groups of three. So AUG CGU is no longer going to be read that way. All right. Because if you add an extra in there, it's going to shift everything over. Excuse me, every amino acid after the mutation is going to be off. If you think of it like reading a sentence, if you say, the fat cat ate the rat, right? But if I delete one of these letters and shift everything over, the fat cat ate the rat is not what you get anymore. what you get is a gibberish sentence. And that is exactly what happens with the protein that's made. So at this point, I'm going to wrap up our first video. And when you're ready, you can proceed on to modern genetics part two, epigenetics, and genetic engineering.